you very much, and thank you for allowing me to present this work here today. Okay, so as he already said, I want to talk about a universal tool for predicting differential expression. So let me start with the background. Maybe you're all familiar with the standard way for predicting differentially expressed genes, or so DAG. There we go already. With DAGs, predicting DAGs in single-cell data. So basically, we start with a large table, right? So every cell is a row, every gene is a column, and we do several processing steps, and we cluster the cells. We visualize them maybe in a 2D plot. So every color here is a different cluster. And then we compare between the clusters. So maybe this cluster versus all the others, and this cluster versus all the others. And we're trying to find genes that have a high expression in each cluster compared to the others, which would be our DAGs, or differentially expressed genes. So there are essentially two steps. So first cluster, and then compare between the clusters. Of course, it makes sense, I guess, but it's maybe not always so straightforward. So because this data is so high dimensional, it's very hard for us to actually look at the raw data to see if our clusters make sense, for example. So here I'm showing you the same plot without the colors. If I would ask you how many clusters there are here, I think everyone might give a different answer. Then also, if I insist that these are reasonable clusters, it's very hard for me to say, why should this be 3 clusters and not 1 cluster, or 2 clusters, or 4 clusters, or 5 clusters? And does the border between these 2 clusters, does it make any biological sense? I have no idea. I cannot check the original data so easily because it's high dimensional. So also, if I have to compare each cluster with all the others, it's time consuming, it's not elegant. And also the DAGs that you can find are somewhat restricted by the clusters that you start with. You cannot get DAGs that don't fit with the clusters. So the clusters restrict the DAGs that you can actually find. So, to make a long story short, it would be nice if we have a method that does not rely on comparing between clusters for predicting DAGs. And that's exactly what we tried to make, of course. So we call our method singleCellHaystack. Okay, so let me try to explain what's the concept behind this method. For more details, I want to refer to those 2 papers. So the concept is we will not look at clusters and compare between clusters. Instead, I want you to imagine some input space. And we are trying to look at where are the cells within that input space. So here I'm trying to show an example in 2D, because this is a 2D screen. But please imagine a 10D, like the first 10 principal components, or the first 20 principal components, or something like that, with all the cells inside them. And here I'm also showing a clear DAG, so it's expressed here but not here, and then a clear non-DAG, which is expressed a bit everywhere. So first, I want to guess, so where are the cells located in that input space? Where is there a high density of cells? Where is there a low density of cells? What I'm trying to show here using the color intensity. Now, this is my reference distribution. Okay. And then for every gene separately, I want to do something similar, but it's not the density of cells, but rather like the density of its expression or its activity within that space. It's almost like what's the density of the RNA of those genes within that space. So for this gene, it's expressed around here. So the dense region would be around here and not here. And this gene is expressed rather everywhere a little bit. So it might look like that. And then if we compare those to that reference distribution, actually this one is completely different from the reference, right? And this one is rather similar to the reference. This is a DAG. So that's the kind of gene I want to find. So it has a biased distribution within that input space compared to the reference. That's the basic concept here. So, and there's no comparisons between the clusters or something like that. Okay, so how do we do that in practice? So guessing that reference distribution is not straightforward, especially if this is like 10 dimensions. So how do we go from here to here? First, we generate many grid points, maybe 100 grid points, that occupy the subspace within that input space where the cells are actually located. And then we're going to scan or calculate the distances between those grid points and the cells using some kernel function and try to estimate what's the local density of cells around each grid point. So, for example, here there are very few cells, so this grid point would get a very low density. And here maybe there are many cells, so this grid point would get a very high density. So that's my reference distribution in this case. So next, for every gene separately, I will do something similar, but trying to estimate the expression distribution. And I'm using the same grid points, but now I'm weighting the contribution to the grid points by the expression of that gene within the cells. So in this case, so this gene, which is a DAG, it's expressed around here. These grid points would get a very high density, and these would get a low density. So I get a distribution like this for every gene, and then I compare those genes, that distribution of the genes, with that reference distribution. And I try to see how different are they. And for that, so we can use something called the Kullback-Leibler divergence. Maybe you are familiar with it. So it's often used to measure the difference between two distributions. So this is the formula which I will ignore here. So let me show, so basically that's it. Let me show two examples to make it perhaps a bit more clear. So this is again my clear DAG. That expression distribution is rather different from the reference. So it would get a high Kullback-Leibler divergence. And this is my gene that's clearly not a DAG here. So this distribution is rather similar to the reference. It would get a low Kullback-Leibler divergence. And then we do some randomization. So we do permutations of the data. We do some modeling. And we try to estimate a p-value for those divergences. So this one would get a very low p-value. This one would get a high p-value. So this is the kind of gene that we want to find here. So we applied this on single-cell data sets. So this is one example from a bone marrow data set. There are about 5,000 cells here. And in this case, so as input space, we give maybe the first 10 principal components. And these are some of the high-scoring genes that we might get. And they are all clearly DAGs. And I should stress again, this is done without comparing between clusters. For this one, we used the R package that we made. And if you want to apply this on a larger data set, like an Atlas-level data set with 4.3 million cells, so for those kind of applications, we also made a singleCellHaystack Python version. So in that case, we gave the first 50 principal components as input, so that's what that data set looks like. And this takes some time to finish, of course, but these are the top scoring DAGs in that case, and they all look like DAGs. So it works on, let's say, normal single-cell data. It works on huge single-cell data sets. But what I really want to stress here is that it's more universally applicable. So I want to show several examples. One is spatial transcriptomics data sets. So I think you are familiar with spatial transcriptomics. So this is one platform. So it's a Visium, 10x Visium platform. So this is a tissue. In this case, it's a liver tissue. For every gene, we can see the expression within that tissue. And there's maybe more than 10,000 genes. And many of them look like this. So it's rather like random expression. But some of them have a very interesting expression pattern in space, so like this one, which is high and low and high and low. So this is what we would call a spatially variable gene. So here I want to predict those spatially variable genes. And in this case, so we don't give those first 10 principal components as input. We can just give the 2D coordinates of those spots as input space. So we give the 2D spatial coordinates to our method. Then it will do the same thing. It will try to estimate the distribution of the spots in that 2D space, and then try to predict those spatially variable genes. And this one would be one of the top scoring genes in that case. So here I'm showing several other examples. So this is one of the top scoring genes in a human liver data set. This is one in a kidney data set. These are 2 mouse brain data sets. So you can see those top scoring genes, they have the very peculiar spatial expression pattern. But you can also use it on SlideSig version 2 data, or you can use it on that HDST data, for example. So I think it generally works. Okay. So how about, so how about runtimes, for example? So we ran several methods on many different platforms. So here I'm showing in the x-axis the size of those data sets. The y-axis is the time that it takes to finish on just 1,000 genes. And actually, many of those widely used methods, they are extremely slow, actually. Our method is not so slow, I guess. There is one method, however, that's exceptionally fast. It's called SparkX, which means, of course, that now I have to claim somehow that we are still better than SparkX. And so here I'm showing a comparison on a 10x genomics dataset, Visium dataset. So I'm showing the log p-values returned by our method or by SparkX. Each dot is one gene. So in general, they rather agree with each other. So many genes are along that diagonal. But there's a subset of genes that gets a very high score according to singleCellHaystack, and not so much according to SparkX. And these are those genes. Of course, it's a bit subjective, but I think they are clearly spatially variable genes. I don't understand why SparkX says that this gene, so this gene, is basically nothing. I'm not so sure why. And on the other hand, so this side of the plot is rather empty, but there are 2 genes here that get a high score according to SparkX, but not so much according to Haystack. And again, it's subjective, but I think these expression patterns are not so striking, let's say. So I hope I can say that maybe our method is more sensitive. Okay, so another application is maybe pathway analysis in some input space. So our method makes very few assumptions, I think, about the input data. So for example, it doesn't have to be read count. Instead, I could say, I will take all known pathways and the genes known to play a role in those pathways, calculate their average expression in all those samples, and then give those average activities of the pathways as input. And then our method can predict pathways that have a surprising expression pattern within those tissues, like these 4, for example. And then another example is trajectory analysis. So the input data could be 2D, like spatial coordinates, could be several principal components, but it could also be just like a 1D trajectory. So here, this is an example on a thymus data set. We predict a trajectory from double negative to double positive to mature T cells. We give that pseudo-time as input. So it's a 1D space. And then our method will predict genes with a changing expression pattern along that pseudo-time dimension. Or you could use it on site-sig data, or you can use it on single-cell attack-sig data, or you could use it on a collection of bulk RNA-sig data. I think it's really rather universally applicable. So as a conclusion, originally we made this method for predicting DEGs in single-cell data. And then we also applied it on spatial data. And so as I just said, you could use it on many other data types. The input space could be 1D, could be 2D, could be 3D, could be 5 principal components, could be 10 principal components, could be 50 principal components. It does not rely on comparisons between clusters. And I think as a result of that, it can find, in theory, almost any non-random expression pattern. It's relatively fast. I think it's somewhat accurate on simulated data sets, at least, although I didn't show it today. And we made an R package and a Python module that you can use. And finally, I should say thank you very much, especially to Diego Diaz, who's also here attending this meeting, and for other people who contributed to this, to people who are funding and might be funding this or similar projects in the future. And also, this is Japanese, but if you are interested in these kind of topics, please contact me anytime.
Oh,
Thank you very much.
thank you, Alexis, for the great presentation. I see there's great interest in the audience. Please present who you are, where you're from, and what your question is. Thank you. Please start.
Okay, thank you for the talk. I'm a math PhD student in the US, so thanks for the very interesting talk. I have some questions about, like, shall we go to the t-SNE part? Like, just use the expression, like the very previous one, like the t-SNE shapes. Yeah.
Please say stop. Here?
Yeah, we can stop here, yeah. Okay. So, like, I think the one thing is that you said the shape of the t-SNE plot does not make any sense, right? Am I right for that's why we like try to find another interpretation for the differential expressions?
Whether the t-SNE plot makes sense or not, I will not make any judgment about it. So those clusters, I should say those clusters are not based on t-SNE, right? And also our method does not use the t-SNE coordinates, but the underlying principal components. I don't know if that's your, I haven't heard the question yet.
Yes, that's one of the questions, like, because we have a lot of like compression methods, right? t-SNE is just one of the compression thing. We can also like use any UMAP or even the SVD or the principal components. Like, does this kind of comparison algorithms influence the result, like for the, because we need to like calculate the KL divergence for the density of the...
okay, so I should stress again, none of those results are using the t-SNE coordinates or UMAP coordinates. Well, of course, you could run it on t-SNE or UMAP coordinates. I'm not stopping you. But so they are based on the, for example, principal components, first 10 or first 20 principal components.
Okay, so just compress the data by the principal components.
Or another similar method.
Okay. Yeah. And another question is that we got like the differential express, shall we go to the next slide?
Okay.
I saw there are slides for the visualization. No, not this.
Let's go through them one by one.
Sorry.
Please say stop.
Yeah.
It's a bit slow.
Oh yeah, I think maybe the previous one. Okay. Yeah, this one. So here we found some like differential expressed genes, right? And did you like try to use this differential expressed gene to re-annotate the cell types? And is there any like different colors like in the very previous plot? Because I see seems that in the very previous, like the very beginning plot, the cell is annotated like use some prior information or?
So did we make a new annotation using those cells, using those genes? No, not at all. So all that I'm showing here is just trying to predict those differentially expressed genes.
Just expression genes.
What you do with those genes after that, I don't know. But here my job stops, let's say, for the moment.
Okay.
Okay, thanks.
Question from the other side, please.
Okay, thanks, Prof. Student from US. And very brilliant talk. I noticed that you mentioned the spatial transcriptomics, right? So you said the tool could be implemented on the spatial transcriptomics as well as single cell data. So in single cell data, we got the cell gene matrix, and you see you use PCA and the KL divergence to mention the similarity between two shapes. And I wonder if you use the same way to the spatial transcriptomics, or you use the coordinates of transcriptomics and do the maybe shape similarity. I'm not sure.
Okay. So I'm sorry, perhaps I didn't stress it enough. So here, so in the previous examples on single cell data, we used typically as input the principal components. And so here, that's not necessary. Of course, you could do it. But here, what I imagine is you just take those 2D spatial coordinates. So we have an x-axis and a y-axis. Give the x and y of each spot or each cell or each beat as input. And then the rest of the method is exactly the same. So it will try to estimate the density of spots in that 2D space, and then it will try to estimate the expression density of the genes within the same space, and try to compare between them and predict the spatially variable genes. So the method is exactly the same, but the input space is 2D here.
Yes, I know. So you see any input space, but imagine I have this condition. So I have a single cell spatial transcriptomics data, which means that we could use the spatial transcriptomics in the single cell level. So I implemented this data on 2 ways stimulatively. First, I use PCA to do the process, and then I use the coordinate to the process. So I wonder if it, in fact, the results, the same data using 2 different input spaces, and we can get the same results of different expression genes. Thank you.
Okay, let me say, please check that first paper. There we made a comparison. So what happens if you use, let's say, the 5 PCs or 10 PCs or 20 PCs? Or what happens if you change some other hyperparameters? How did the results change?
Thank you. I think we need to give the other questioners also a chance. Please, from the left side now.
Okay, thank you for your work. I appreciate that we are able to call DEGs even though we are blinded to clusters. So my question is, I may have missed it, but how did you choose the nodes? And because I believe that they will influence the reference distribution and eventually affect the DEG calling.
Yes, the question that I feared to hear. Okay, so I have to apologize already because I said the whole time this is clustering independent. It's all a lie. Because how do we pick those grid points? Actually, we use k-means clustering. I don't care about the clustering result at all. What I want to do is I want to fill that subspace where the cells are with grid points. So it's a kind of a space filling problem. I tried several methods for doing that. I want those grid points to cover the subspace where the cells are uniformly. I want them to be not all next to each other, but uniformly spread. I tried several methods for doing that. But the best method so far is, unfortunately, use k-means clustering, get the centers of each cluster. Those are really nice grid points.
What means best in this context? I think the questioner would like, what is best here?
The grid points will be close to the cells. So this space is actually very empty. There are no grid points here. There are no cells there. This is where the cells are. There is where the grid points will be. Also, the grid points, they are not all like clustered around here. No, they fill the whole subspace where the cells are. So that's what I want for the grid points. Those are good grid points. Unfortunately, it's based on clustering. Okay. But we don't compare between the clusters. Yeah. I don't care about the clustering result. And if someone knows a better way to do this, please tell me. Yeah. Okay.
Next question, please.
Hi, thanks for the great talk. So I just want to ask about the expression. Actually, did you filter for the expression as well? For example, in here it's just like a presence or absence of the density, right? And then what happens if there is a much higher expression for one side of the 10-dimensional space, and then the other one, actually they are also expressing it, but at a much higher, lower level. I mean, density in the sense that, is it a 1 and 0 and 1? In the sense that if they're expressed, then you consider them for density analysis, or do you also take into account the expression for 5? Because some cell can express 5 and then the other one 10. And in the conventional DEG analysis, we will pick them up, but will they pick also in here?
I'm not sure if I understand the question, but so in the version that we have now, so it's the densities of expression is weighted by the expression level. So it's like a continuous contribution that each cell makes.
The density is weighted with the expression level.
So it's not zero, or it's not expressed or not expressed. Maybe it's better to discuss about it.
Okay, we can discuss it later. Thank you.
The last question, please.
My name is Sorin Dragic. I'm a program director at the National Science Foundation in the US. This is fantastic, okay? I'm a computational person, and I always thought that it's kind of crazy to have a bunch of clusters and start randomly comparing them. So I really appreciate it. It's a very elegant solution. However, the life scientists that I work with, they do something a little bit different. They do some kind of t-SNE or whatever, and they identify cell types based on some gene markers. And then they want to look at differentially expressed genes between cell types. So, and it's possible that several cell types have the same pattern of expression for any one particular gene. So if you apply your approach, all those would be differentially expressed, but not between cell types, between that group and something else. So have you had any interaction or any feedback from the life scientists as to how would your method work with cell types in that context? Does it make sense what I'm saying?
Thank you, first of all, for your interest in my work. Secondly, I know it's a kind of a losing battle, actually, because people really like clusters. And they like to, and I also like clusters, and I like to assign the cell types and then compare between the cell types. I don't know.
So for instance, have you made any comparisons between the differentially expressed genes that you get with your approach, which are kind of differentially expressed between this area and that area, right? Any areas, right? And the ones that would be obtained, sets of differentially expressed genes that would be obtained by comparing cell types based on markers?
At least I guess I should refer again to that first paper.
Okay.
Where we compared our results with those of like the typical cluster-based results.
I'll read them both. Thank you so much. It's great work. Thank you.
Yeah.
Thank you very much.
Thank you